Hello, I am Beatrice Salvatori, and I'm a scientist from Negedia. Hi. Thank you for being here. So I'm going to talk today of how we have applied transcriptomics using machine learning in order to try to bridge the gap that is today present for the translational application of NGS in clinical practice. So Negedia is a company that was born around 5 years ago. In 2020, this company was a spin-off of the research center of Telethon Foundation, and we were able to provide, from the beginning, NGS services to all life sciences. But soon we optimized and we improved all our workflows in order to provide clinical level NGS services, and we are today participating in several clinical studies where we are able to provide NGS services. But the reason why we were born is because our mission was to enable precision medicine, and so we wanted to take advantage of the know-how in order to transform the cutting-edge sequencing technology into accessible and interpretable clinical solutions. And so today we have developed several tools. We have 2 patent pendings, 3 patents that were obtained in Italy, and 1 publication that is currently under revision that for which I'm going to show you the results today. So the most important thing that I want to start with is the translational gap, so that gap that we found that exists between on the cutting-edge technology in the field of NGS and their actual application in the clinical field. And we found some reason for this. I mean, obviously the reasons are always very complex, but there are some main reasons. Starting from the very high NGS cost that is not feasible for the today clinical practice, the clinical validation that is needed in order to use a tool in the clinical field, the technical expertise that is required to handle NGS in every pathology lab or any lab that is linked with the patient, and the bioinformatics that is also needed for the analysis of the data and the interpretation. So we started a study, as I said, trying to solve and do something to shorten this gap, and we got some funding for which we were able to carry on our research. I'll start with the field of molecular pathology, where we found a lot of challenges that mostly are linked to the running of the precision oncology and all the requests that are coming from the field of precision medicine to the pathology lab. And so, as you know, the pathology labs are based with the study of immunohistochemistry with individual markers. It's this type of method. It's very robust and informative, but it's limited to the number of markers that can be done in the tissue with the tissue that the pathologist has. It's totally operator dependent. We know that IE is solving a lot of this issue. But still, from one piece of tissue, you can just analyze few markers. Instead, the NGS is totally another approach. So from the tissue, you can have more than 10,000 RNAs that potentially can be the markers, and you can get a digital diagnosis. It's scalable, comprehensive, automated. But the point is that all the technology that was available so far was not able to be actually applied in the clinic. So we took advantage of technology, what we have set up and optimized in our lab that we call 3' digital gene expression, and I will show you the details of this technology in the next slide. This technology, this is very important, is totally cost effective. So the cost of this technology is around 1 assay of immunohistochemistry. It's, I will show you, we validated on clinical samples. It has very few steps, so it can be easily transferred to a pathology lab where the molecular pathology usually do some type of other QRT, PCR tests or so on. And it's automatic, and the data, it's digital, so can be interpreted through machine learning, as I will show you. So we start with the clinical samples like an FFP. We just need a couple of scrolls of the FFP block. We extract total RNA. This RNA is very degraded, so it's terrible quality, but this doesn't matter because we apply this workflow. So this RNAseq library that has the peculiarity of being a library that is focused just on the 3' end of the RNA. So we don't sequence the whole RNA, just the 3' end. So just that region that is very preserved because it's usually covered by the poly binding protein. So we just sequence 100 base pairs. We just need 8 million reads, and this workflow is very cost effective. It's totally simplified. I can go on any sequencer. Even a mRNAseq can load around 10, 20 samples. And so we validated also, because we knew that this type of workflow was very robust, we validated the comparison of fresh samples with FFP samples that, as I said, have nucleic acid that is really compromised in HeLa cells, but also in tissues. And so we prepare a library from both type of samples. The tissue we just took, we took a tissue from 7 patients. We split them in half, and then we analyze fresh and FFP and the tissue included in FFP. And as you can see here, the correlation between the 2 library preps, it's totally comparable. So, and also in terms of number of detected genes, so quality, the number of unique reads, we have very comparable numbers. So we have around 12,000 genes in HeLa cells and even more in the tissue. We also decided to test the precision of this technology by checking for repeatability and reproducibility. And so here we were very surprised and very happy of these results. So for repeatability, so just doing 2 libraries in parallel at different times with 2 different operators from the same piece of tissue, we have 93% concordance on the whole transcription. So we're talking here about more than 12,000 genes. And the same result, it's very comparable and very good for reproducibility. So after doing this, we decided to expand our study and collect more samples and try to validate this technology. So the RNA analysis, the RNA study on a wider cohort. So we built a data set made of different cohorts. We also collected samples from normal adjacent tissues coming specifically from the PM cohort. And so we started to analyze more data. And as you can see here, also we checked the failure rate of our library prep. For RNA-seq is usually really high with samples that are older than 6 years, but here we have a 0% failure rate on samples that have just been prepared, younger than 1.5 years, and then we have a 4% failure rate for samples that are 6 years and even older. They go up to 10 years. So this is a technique that is really good for archival samples if you want to do retrospective studies. And then we checked also what was going on, if we had any batch effect. So what you see here, a plot of a U-map. So every dot is a sample, and the more proximal are the sample, the more they are similar in terms of whole transcriptomics in their transcriptional gene expression. As you can see here, there's no basically effect on the cohort or on the running date. The only difference that we see, and I will show you on the next slide, it's the difference of the different types of tumors. And so going in detail in this data, here we have mapped the tumor samples and the normal samples, so the samples that are adjacent to the tumors. And as you can see, they're totally different in terms of transcription. And this really tells us that transcriptomics has all the information we need to distinguish these type of tumors, these type of samples. Also, there was a number of samples that were unassigned, so misassigned by the pathologist. We obviously clearly saw that through transcription, and then we did a secondary evaluation through the pathology that actually gave us, told us that we were right, and the samples were, in some cases, hard to be analyzed by hematoxin eosin. And so we reassigned these samples, but then we thought, okay, now we can see all the biobank, it's beautiful, but if we just get one sample, how can we distinguish if this sample has tumor cellularity and is actually eligible to do any biomarker analysis? And so we compared two different types. So we compared in our data set samples that were tumors with samples that were normals, and we actually applied a feature selection algorithm in order to find a signature of genes that could actually be representative of this difference. And as you can see, this is the signature. We have completely reverted the gene expression pattern of this signature from 0% cellularity to 10% cellularity. So then we applied a GLM model, machine learning, in order to have a binary response. So given this signature, we applied on the sample, and we just get to know if that sample has tumor cellularity or not. And so this is just the output. So is the sample adequate for do other biomarker analysis? This is the testing accuracy. As you can see here, we got 98% accuracy with specificity of 100% and sensitivity of 98%. So there were 4 samples that were not actually assigned correctly. If we look at them and go back on the whole transcription, as you can see here, they're always in the same part of the UMAP where the pathology also got mistaken. So there's something weird in the samples, and we were happy we were not putting them as considering them as tumoral. We will do more analysis, more in deep, to have more information about this. So then we said, okay, now why don't we just validate this technology also on the pathology lab, so on the clinical setting. We didn't want to reinvent the wheel, do new biomarker analysis. We just took those biomarkers that are working very well and are segregating and classifying very well the patient in high risk and in good prognosis and bad prognosis for breast cancer, so in the molecular subtypes. So what we did, we just plotted, so we just took the level of RNA of these genes and plotted them again in a UMAP. As you can see, the 4 markers that are usually used in the bioimmunohistochemistry, also by transcription, are able to segregate perfectly the subtypes of the breast cancer. And so then we said, okay, let's just transfer the information that we have on positivity for these markers on the transcriptional levels of these markers. And so we again applied machine learning, and we found some threshold. And so what's the output? The output is just a range of expression of that marker, and we know where to put the positivity. So we just need to get a sample, we measure the level of the RNA of these markers, and then we say, okay, this is positive, this is negative. So again, it's a binary response, and it's really what the clinicians need. And here the results are surprising. We have a 99% accuracy compared to immunohistochemistry. So we are very close. But what we have more, we have other 12,000 genes that we can use for precision medicine to test any other biomarker. I just want to go back in what you saw before. So the markers are pretty good. There's only one marker that is not working perfectly, and it's the MKI67. The MKI67 at the level of transcription is not a good transcriptional, it's not good for dissecting, for segregating to the subtypes of the hormone positive that are luminal A and luminal B. So again, we did our feature selection, and we found that 30 genes made better resemble the differences between hormone positive proliferative and non-proliferative in this group of patients. As you can see here, this is the evaluation of a pathologist. Our gene expression signature better segregates the 2 subtypes, so the light green with the darker green. And so, again, the result is just binary. So we have samples that can be highly proliferative and less proliferative. And so, as I said, so this is just the beginning of this, because we have other 12,000 genes that we can analyze. And I'm just reporting here just few examples of what we can do with all this information. So, for instance, we have done the analysis of HRD tumors. In this case, we're not looking at the mutation levels, we're not looking at the DNA, but we're just looking at what happens at the transcriptional level. So we took advantage of an old paper that seems to be very robust, but was never where they identify a gene expression signature, but they were never able to put this in practice because, as I said, RNA-seq was never considered a good tool for clinical samples. And so we applied this to our cohort, specifically on triple negatives. And as you can see here, we were able to distinguish two clear subgroups as a group that was HRD positive and a group that was non-HRD. And when we look at the progression-free survival of these samples, as you can see, we obtain really what we expected. So these patients treated with chemotherapy have a better response if they are HRD positive. And this is what is known because HRD induces synthetic lethality in these patients. Another thing that is very interesting that we can do, and we also had a patent on this, we can measure the HER2 activity. So we know that ADCs today are working very well to inhibit HER2, and not only doing that, they can also be internalized and they can have an effect on the cells. But what you need in order to have these, it's HER2 present on the membrane because it needs to be bound by the antibody. So why don't we, instead of just measuring expression that could not tell us actually if the receptor is on the membrane, why don't we just measure the signal transduction pathway? So we have 12,000 genes to do that. So that's what we did. We found the genes that were going up and down based on the HER2 activity, and through our transcriptomic approach, we found 46 genes that represent the signal transduction at the gene expression level. And this is something very interesting. We're plotting here breast carcinoma, but also other types of tumors. This is our data set. The other ones are TCGA data set. And as you can see here, there is a relationship between HER2 expression and HER2 activity that is sigmoidal, in which not always when HER2 is highly expressed, it means it's present on the membrane and the signal transduction pathway is active. So it means you can have HER2, but maybe it's not on the membrane and maybe it's not working. So our activity measure is able instead to tell us if HER2 is on the membrane and it's active. So just to conclude, we have made several modules to apply our 3' DGA approach. But what is very interesting is that you can study any new biomarker. You can add up signatures. It's just one test. We just need few slices of the FFPE. And this is the specificity and the sensibility, so the accuracy that we have compared what is the gold standard today, that is immunohistochemistry. This is just an overview what we can get in the result. It's very similar to what pathologists are used to observe. If you have any question, please ask. I hope, but it was not complicated. Thank you very much.